Good morning. Thank you all for coming in on this bright and sunshiny spring morning to the third section of Physiology 452. I'm Dr. Rasmussen. I'm in the Biomedical Research Building, room 360, and I will be teaching the next section all the way up till the final. I have a few different policies than went on before, in that we will not be using the interactive modules. There will be no extra credit. So your grade will depend on your performance on the test alone. I'm happy to answer questions. I've found in the past that email questions, unless they're very, very simple, I in general will not be able to answer them because all too often either I've misunderstood what the person has told me or my email back has not been clear enough on the subject. And so it leads to a lot of confusion, but I'm happy to talk with anyone about any questions they should have on the course, about the material, and about what it means. There is one question or a class of questions that I will not answer. And given the size of the class, I have to say this every year. No offense intended to anyone in this class, but I'm not going to answer the question, do we need to know this for the exam, or is this going to be on the test? And I'm always amazed at the creative ways that students come to ask that question every single year, but I won't answer that question. And the reason for that is fairly simple. You're here to really get an education. That's more than just simply memorizing the bare minimum you need to get by. So, and we can only test on a very limited sort of thing. So you have to kind of guess what's important and what's not important. We are here to guide you through and give you a sort of an overview of what is the main and important things. I'm not going to leave you completely bewildered as to what's going to be on the test. In fact, I try to make it as easy as I can to show you what's important. If I say, this is an important term, or it's up there bold and in red and underlined, that's a piece of vocabulary or a concept that you really probably has a high probability of appearing on the test. And so keep an eye out for those. They mean something. I don't do those simply to add excitement to the class. It's to add sort of an emphasis that this is one of the key concepts. Similarly, as we go through and we learn things, people ask about the book. And are you responsible for the materials in the book? And that answer is yes, even if I don't cover them here. And my opinion on that is, of course, you can guess if I'm going over it in class and taking the time to explain it, that's going to be the more important material. But we're here to try and get you an education, more than to learn the bare minimum. So we do test out of the book and stuff that is in the book that is not particularly discussed in class. Material on the book, if I don't cover it, you may see it has a lower probability, and some of the stuff in there is probably way too complicated to even ask a test question on. But part of learning is, and an education is distinct from memorization, is learning how to sort out information, categorize it, realize what's important and what's realistic. And it's a skill that will carry you through the rest of your lives. So that's the way things are set up. That's the reason that I set them up this way. There's a slight deviation from the syllabus. I'll be giving a series of 7 formal lectures. The 8th lecture I reserve in case we don't get through all the material, in case there are a lot of questions or such, in which case we will continue those lectures on into the 8th lecture and complete them there. And that part will be recorded for future instruction. But the main thing about the 8th lecture is there's also right before your exam, and it's at the end of the year, And so I reserve that portion for a review session. So you can come in, and it's completely open format. We do the chalkboard thing. I encourage everybody to come down front. But because I find it's very inhibitory to really discourse about what you know, what you don't know, and how to ask questions, and how to get frank answers to questions, to have a videotape running for that sort of process. So that will not be taped on that 8th lecture. And I'll also warn you ahead of time, that 8th lecture, I will be leaving shortly after that to go to the national meeting of the Organization for the Study of Sex Differences to give a talk. And so I will be unavailable most of the days, the 3 days right before that exam, or 2 days right before that exam. So get your questions in, find out what you don't know, and either do it on that Tuesday during the class or try and do it ahead of time, because I will not be here the day or two after that in order to give you answers to your questions for the test. After the test, we of course go into the finals and reading period, and I will have another review period. So once you get your tests back, if you don't understand why you got certain things wrong or what was wrong, or you want to prepare better for the exam, then I will set up during the reading period a review session. When and where that's going to be is still yet to be determined. So sort of the organization of my portion of the course, and my portion here will start with muscle. And muscle is, of course, very important in your body. It does a lot of different things. Generally, as physiologists, we characterize muscle in 3 classes: skeletal, cardiac, and smooth muscle, each one having very distinct properties and organization, and really very distinct functions. And since this is supposed to be the cardiac section, of course, our emphasis will be on the cardiac muscle for most of it, but it's important that you know why cardiac muscle is unique and what these other muscles do and how they function differently than cardiac muscle and why. So what does the heart do? Structure and function is really the core of physiology. So your structure really is optimized by evolution to your function. And the heart does really one thing. It's a really important thing, because if it stops doing that, you're dead. It pumps blood. And it does that, and it's a sort of a 2-stage pump. So we have our atria, which are little reservoirs here that collect your venous inflow. They contract and squirt a little bit into the ventricles. And the ventricles are the workhorses. They're the ones that actually provide most of the force behind the blood. So sort of a 2-stage pump. A little collecting area with contractile behavior that's coordinated. And then your main ones, which are your ventricles, one going, of course, to the pulmonary circulation, to your lungs, which is separate then and at a very different pressure than the one going out to your aorta and the rest of your body. So a heart pumps blood, pretty simple, and it does it in this 2-stage process. So what do physiologists like to do? We like to look at things quantitatively and as physical phenomena. So physiology you can think of as a contraction of physics and biology. And so to see how hard a heart is working, we look at, and what its function and dysfunction might be, we look at something called a pressure volume loop. You'll see these again later, but just to introduce you into the idea of going from a physical process that's occurring like that onto a graph, and understanding things. So when we start, we have to start somewhere. A heart is rhythmically beating, so it's going in a cycle. So we'll start at point A here, and this is where the heart has just finished relaxing and is about ready to fill its chambers. And you see here that it's opened, and as it is opening and still relaxing, blood starts to come into the various chambers through the atria and into the ventricles. You see this valve here? So it's sort of a one-way valve. It's open. They're both flowing. They're pretty much at the same pressure. You see this reduction in pressure as this is relaxing and allowing things to go in. As you go further, then that relaxation process stops. The pressure of the return flow of blood starts to fill things, and you get to this point here. So both the atria and the ventricle are kind of full at that point. At that point, you have sort of the order in this two-stage process. The atrium then contracts and gets this last little bit of blood in here, as much of it as it can, out of the atria, and kicks it in there. So you get this dicrotic notch here, which is one of the sounds you can actually hear in a stethoscope, particularly if you've got valve problems. So you have this little notch here, things close down, and then you have a straight line up. So on this axis, you have volume. So as it's filling, you're going and you're filling up the volume here. So these things are going from empty to distended, and you're at a very low pressure the whole time. So now this valve has closed, and this valve is closed, and the heart starts to contract. Well, water is what's known as an incompressible fluid. So it's not like a sponge. When you squeeze on it, it doesn't get smaller. It just stays at the same volume. And so you have what's known as an isovolumic contraction. So your heart muscle starts contracting, and it's contracting harder and harder and harder. And the pressure in that chamber then, this is pressure, starts to go up and up and up in the ventricle. This is the pressure of the ventricle. So it starts to go up and up and up. And as it's contracting, what finally happens? Well, the pressure between the chamber of the ventricle and the pulmonary artery or the aorta becomes equal. And in fact, eventually becomes greater than the pressure in the pulmonary artery, and this little door here, it swings open, just the way it would if you were pushing on a set of swinging doors, and the blood starts to flow in. And as the blood flows, you are no longer having an isovolumic contraction. You're still contracting, you're pushing that blood as hard as you can, but the volume of the heart is changing because you are actually removing that incompressible fluid, or pumping it out into the aorta. And that goes on for quite a while until you finally have contracted so far that each additional contraction is not really generating much additional force. You've pumped out most of the blood, and you've gotten back then to your resting volume. That is, there's no more volume there really to be pumped out. So when that happens, your heart is relaxing, because you can see the force relaxing here. And when that pressure exactly equals the pressure in the aorta, that valve shuts again. And so when that valve shuts again, you get what's known as isovolumic relaxation, because you're at a fairly high pressure up here. Your venous return is at a very low pressure, and so everything has to start filling. It can't fill because you're still at higher pressure, so you have to relax, relax, relax. Once you start to relax far enough that you're down to very near atmospheric pressure, everything starts to fill again, and you go through the cycle again. Now, you may remember from physics, force equals, a force through a distance is equal to work. The same set of units work for pressure times volume is equal to work. And so the integral of the area around this curve is the pressure volume work, or the stroke work that occurs in the heart. And that's why it makes a fairly good indicator of how the heart's working. So we have different chambers here in the heart and different valves functioning at different times to operate as an efficient pump. Now, controlling that pump is important, because you have different demands on the heart. You know, when you're sitting here, your heart isn't doing much. When you're out, say, jogging or something like that, your heart is doing quite a bit. Now, a lot of that is neuronally controlled, but the heart also has its own intrinsic system. And this is a pattern you'll see quite often. You may have sympathetic control, getting your heart going faster and pumping faster, so from neuronal inputs, but at a lower level, you've also evolved another thing. And so you have multiple feedback mechanisms that will enable the organ to adapt under a variety of situations and at a variety of different levels. So the heart actually has a system by which it can adjust the strength of its contraction and its output in relationship to how much blood comes in. And this is what they go on in your book about as the Frank-Starling law, a term you should know. And it basically says that over most of the physiologic range, let's not worry about this up here. Something's starting to break down. If you stretch things far enough, they stop working. But over most of the physiologic range, when you're at diastole, or diastole, your heart is relaxing and it's filling. And it's basically a limp bag, but it's an elastic bag, so it's like a balloon. You know, the more pressure you put into it, the bigger it'll get. And so it stretches the myocardial fibers that are in the muscle, and this stretch of the myocardial fibers sort of has some resistance to it. This is elasticity. And so it's elastic, it stretches up to a certain point. And in heart muscle, if you stretch the muscle ahead of time, then the more you stretch it ahead of time, the harder it will contract. So if I've got a little volume and I'm pumping a little blood, I don't develop much force, and our pressure volume loop goes around here. As we go up further, we're filling up a lot of blood, we contract really hard, get that blood out of here. So there is a relationship between your resting stretch, or your resting heart volume, and how hard it's going to contract. Entirely a property of the cardiac muscle. This is not mediated by your nerves. And so as we go through this, we're going to talk about how and why these things develop, and what are the things that, you know, I mean, this, how and why this sort of thing would develop, and what it means to regulation. So here's another one. If I emphasize it, it's likely to be on the test. So you have a heart, and it's a very specialized type of muscle. It's got one task to do, but it's absolutely essential, differentiated very early in evolution. And to understand how it's different from the others, we're going to look at and know the basic muscle types and properties. The muscle, of course, that's most abundant in our body is skeletal muscle. Skeletal muscle is what we think of as muscle. It's under voluntary control. It's the things that determine our motions, balance, and such. And it in and of itself is not a single muscle type. We have slow twitch fibers, fast twitch fibers, and they're specialized for various tasks, such as strength or speed. So, you know, you have the white meat on a chicken. That's really designed for flight, high energy output very quickly. And you have the darker meat for running, sort of a very different sort of task, requiring different levels of strength and different oxygen consumptions. But the basic organization of all skeletal muscle is sort of the same. So you have a skeletal muscle, and you look at it, and, you know, you'll see a flank steak or something like that. It's really obvious. It's got these stripes in it. It's organized in little bundles. And so at various levels, you have the skeletal muscle. You cut a piece of it. You look at one level, it has a fasciculus, and a fasciculus is a bunch of fibers. So the fibers organized into bundles of fibers, and then we have these muscle fibers, which is essentially one big fused cell. So the fasciculus is many cells, many of these fibers put together that can be recruited to cause contraction. We have a fiber, which is basically one big cell. Within the cell, we have separate units of organization. Again, sort of almost like repeating on various levels. You have a myofibril. Now, a myofibril is a subcellular level of organization. And these are where the contractile proteins are. You have them organized into these stripes or striations. This is why skeletal muscle is frequently called striated muscle. A striated muscle has this organization of sarcomeres, where you have a Z line where all the contractile apparatus is connected from sarcomere to sarcomere. You have a thick filament and a thin filament. And these slide against each other to produce force. So it's the sliding of thick filaments on thin filaments. Again, there are several terms on here that you probably should know. Thick filament, thin filament, myofibril, fiber, and what they are. Because they look very similar, and it's easy to get them confused. Because you got a fasciculus. Well, if you look at the picture of the fasciculus, it really doesn't look any different than the picture of the fiber to look at it. But remember, they are at enormously different levels. So a myofibril is that highly organized array of contractile units. All right? That's basically a subcellular item. A sarcomere. A sarcomere is that basic contractile unit. It goes from one Z line to the next Z line. So you have that in there. And this organization, you have a fiber, and that's a large, basically a big cell. It's a large multinucleated array of myofibrils, essentially one large fused cell. And in fact, that's basically how they form. So you have a lot of nuclei, a lot of mitochondria and such, and you have organizations. So starting down at sort of the lower end of the organization, what's in a myofibril? Well, we've got proteins, some big polymeric proteins. In a sarcomere, we have several lines that can be distinguished anatomically. And you look at these lines, and of course, they are formed by either things that sort of connect and anchor these proteins together, the M line and the Z line. And then you have the A line and the H line, which are from the overlap of these polymers, or of the actin and the myosin, where they slide together on each other. So you have these myofibrils there, and they're what actually contracts. They have a regular structural organization with some proteins. They're very small, as you can see here for the scale. And then we have something that amounts to a control apparatus for them. We have the sarcoplasmic reticulum. Now, I assume everyone here has heard of the endoplasmic reticulum, which is, of course, where protein synthesis occurs, and some of you may know that it's involved sometimes in calcium homeostasis and signaling. Well, sarcoplasmic reticulum is highly specialized, particularly for the uptake and release of calcium. So it has a longitudinal component, that is, it goes along the edges of the fibrils, and then it has these terminal cisternae, which are filled with a protein that is called calsequestrin, and serves as really an anchor of calcium. And then we have T-tubules. Now, what are T-tubules? It's a fairly confusing sort of process. This is a structure that delves deep into the cell. It goes basically in heart cells sometimes all the way through the cell. It goes deep, deep, deep into the fiber, and but it's part of the cell surface membrane. So it's basically, if you look at the membrane, it goes down, and then it folds in here, and it carries, it's a little hole, a wormhole, basically. It goes down into the cells and abuts this terminal cisternae. And the purpose of that is to get the signaling from the cell surface down to your contractile units, which may be buried very deeply within the fibril. So you really need to know what the sarcolemma is. The sarcolemma is the cell membrane. So that's the word we use for that cell membrane that covers the extracellular portion of it. Not to be confused with the sarcoplasmic reticulum, which is, like the endoplasmic reticulum, an intracellular membranous structure. In its highly specialized form, adapted for very rapid calcium uptake and release. So it's taking up calcium. The T-tubules are portions of the sarcolemma that dive basically deep down into the cells and provide cell surface area way down that's electrically connected then with this way down into the cell to provide rapid signaling so that you can contract an entire muscle fiber fairly uniformly. The terminal cisternae are specialized structures that hold that calcium. They have calsequestrin in it to store them, and you need to do that. So you need to know where the T-tubules are, the terminal cisternae, and the longitudinal SR, which is really more oriented towards uptake or reuptake of calcium as you go through the calcium cycle. So T-tubule invaginations, that's the cell surface membrane. Sarcolemma, cell surface membrane. Sarcoplasmic reticulum, intracellular membrane, not connected with the extracellular surface directly. Terminal cisternae, part of the SR. Longitudinal SR, also part of the SR, or sarcoplasmic reticulum. We will refer frequently to sarcoplasmic reticulum as SR. That's just jargon that's commonly used in the field. It makes things a little bit easier than having to write that out, particularly on every slide. So I've gone on and on about calcium. So why would I be particularly interested in calcium? Well, I've alluded to the fact that calcium is a signal. And the signal goes from electrical to chemical to mechanical. So you have an electrical impulse from your brain. It initiates an action potential in the membrane of the sarcoplasmic, no, not sarcoplasmic reticulum, in the sarcolemma. Okay, so it's coming into the sarcolemma. It's an action potential. Same as a nerve action potential in many respects. It's that which you covered in 451 action potentials, correct? So same sort of thing, a different shape and a different time course, but that same sort of thing. It's an excitation, it occurs in an all or none event, goes through the sarcolemma and excites the cell membrane. Well, that excitation of that action potential, you can see here on this set of events, flows along the cell surface. In order to get down into these signaling things, this cell surface, which is also a capacitor, remember, membrane anywhere surrounded by electrolytes on either side is a capacitor, so it charges up that part, and in charging that up, it depolarizes the T-tubules. So you get this rapid electrical flow. It's shunted into the deepest portions of the cell so that you can get a uniform thing. Now, we have here what's called a calcium release channel. The interesting thing about a calcium release channel in the skeletal muscle distinguishes it from cardiac muscle is that it's actually directly coupled to the sarcoplasmic reticulum ryanodine sensitive calcium release channel. So there's actually a physical coupling between the motion of the dihydropyridine receptor, dihydropyridines being a class of compounds that selectively block these types of calcium channels, causes a movement of that channel, couples here to the ryanodine receptor. The ryanodine receptor then dumps cell calcium out. So it's dumping cell calcium out. And this calcium has to diffuse out of that limited space here. So the SR is connected by proteins, not by membrane, which membrane is lipids. This is proteins that span this. It's connected here. It transduces the voltage here directly across this gap physically to open up the ryanodine sensitive channel, or the RYR, or the calcium release channel, or sarcoplasmic reticulum calcium release channel, and that calcium comes out. It has to diffuse out into the cytosol. And it has to diffuse there. This is one of the reasons we have this whole distributed network and broken into little chunks. Diffusion is a relatively slow process if it has to go over a very large distance. So having this specialized structure all over the intracellular space gives us fairly short diffusion distances. It binds to troponin, and through binding to troponin, it activates contraction. So you have action potential, excites things. Ryanodine sensitive calcium release. Ryanodine is a poison from a plant in South America that works through opening these particular channels, causes you a great deal of rigor. And so you open it up, calcium comes out. This calcium takes time, so you see these delays here, to diffuse, bind to the troponin, which then takes additional time to actually move that mechanical network into force. So we go from electrical activation via the action potential to chemical activation of calcium. And from that chemical activation, we go to the myofibrils and generate force. So these subcellular controls work in a coordinated fashion. Now, the longitudinal SR has a calcium pump, or SERCA, the sarcoplasmic reticulum calcium pump. And it is like that, the ATP-sensitive sodium-potassium pump that occurs on the cell surface. It uses energy from ATP and recharges the sarcoplasmic reticulum with calcium. And it does it through that longitudinal SR, so that it's, because it's diffused out, you need a wide collecting range. And so that longitudinal SR, as it goes over the length of the entire sarcomere, can pick this calcium back up and bring it back into the longitudinal SR. It has to diffuse down to the terminal cisternae, where it's ready to be used again. So this is sort of the calcium cycling internally that occurs every action potential. Action potential, spread of excitation, release of calcium, cycling back and forth. And that's every cycle you go through this. So it's important to remember, and that's why these emphases are here, that T-tubules help spread excitation from the sarcolemma down into the deeper regions of the fiber. The calcium release channel in skeletal muscle is a direct signaling mechanism. So there's a direct coupling between the SR and the T-tubules. And the reason I keep emphasizing this, you may find it strange, because one of the things that distinguishes the cardiac muscle is it operates by a different mechanism there. And basically, for most, all of the muscle types, calcium is the signal that initiates contraction. So calcium signaling is key. So we've talked about calcium going in and activating things. We've seen sort of where this myosin-troponin complex is in terms of the Z lines and such on the sarcomere. So this is a cartoon representation from your textbook of exactly what is going on. And you have myosin, actin, and troponin, and tropomyosin. So when you have calcium come into the skeletal muscle, it has to initiate events. What it does is it binds to troponin, or troponin C on that. That in turn moves tropomyosin. So you got this little thing there, it exerts some force on the tropomyosin, that moves things away so that now the head groups here on myosin can interact with the actin. And you see how these things are drawn sort of like golf clubs sticking up? You can think of them as little arms. And so they'll contract like this. And each little one of them, when they become activated, will pull. It's not always very obvious from some of the pictures in the book exactly what that is. So remember then, so they're these filaments that slide on each other. And when you were looking at that Frank-Starling law curve, you can see why it would occur that if you have some stretch on the muscle versus no stretch on the muscle, you're going to get different levels of contraction. Because if the myosin, if the muscle is, you got no tension on it, it's mostly contracted, then the overlap between these myosin filaments and the troponin filaments here, this overlap here, you'll be down here and down here. There's not much of anywhere to go. You can't develop much force. As you pull these apart, then you have a longer distance to travel. You can develop more force. And as you can develop more force, you have, you have a longer way to go, so you can develop more distance. That means more force. If you go too far, you start to have too little overlap with them. And so that's where that breakdown is. And you don't want to stretch your cardiac muscle that heavily. But they're in these overlapping things, and you can appreciate then that the longitudinal SR, if it has to go into all these regions here and diffuse back, you want the longitudinal SR to be able to collect that all back up, and then you want the T tubules and the terminal cisternae to be basically dumping where that overlap is the maximum, so you get started right away. And so you can see there's some organization to this thing. So, but we have these thin and thick filaments. The thin filaments have actin, troponin, and tropomyosin. So in spite of being thinner, they have more named things in them. The thick filaments contain myosin. And contraction and relaxation consists of sliding these filaments against each other, and in that movement of that myosin head group as it pulls on the chain of these things here. So, calcium. Where does calcium come into this? Well, you need calcium there, and you need a lot of it. So you have your calcium binding group on your troponin connected to your tropomyosin. You have your myosin head group. ADP is here. It's all ready to go. And it's binding. That's because you have 4 calciums attached. 4 calciums attached causes conformational change, and it's moved that thing just this. It's very hard to see from this what they're trying to do, but you see how it's moved a little bit from here to here. So it's sort of slid it around that thing and out of the way so that the tropomyosin head can bind. And then if you look at then the amount of force you'll develop for a specific preload and afterload, that is specific tension maintained on the tissue, you find that it's a very steep curve. As you go from one level of calcium up to another, you have to have 4 of them there. So it's like a hill plot with an order of 4 at it. So you get these calcium in, you need quite a lot of calcium, but once you have it, it just rapidly goes. So you get almost an all or none response, or a very steep type of concentration dependent curve. Because it's 4th order, you have 4 calciums. And what it's doing is binding things here, binding here, and pushing this away from this head group so that it can interact. Otherwise, it has a weak interaction. Once it has that strong interaction, it can pull. So, what is the cycle that we go through for the cross bridge? Excitation and relaxation. When you're sitting at rest, you don't have any calcium there. So you're sitting there, your myosin is sitting there, it's primed, it's ready, it's got the energy bound to it, and it's ready to go. And it's just waiting to interact with the actin in order to do that. So you get your calcium in, it binds, it moves things out of the way, that cross bridge attaches here, and then it loses the ADP and the PI, that energy that's already attached to it and ready to go, and boom, it goes through a conformational change. See, it sort of kicks or flexes and pulls the chain this way. And so when it's finished doing that, it will remove. If there's still high calcium, it'll go right through this and it'll keep doing that. Just keep pulling like this. And if you think of millions of little hands sort of all pulling their little bit on the muscle fiber, that's what generates tension in your muscles. If you are so unfortunate as to be dead, you get into this state. You have no more energy, so you can't relax this and unbind from this to reprime. So because you have no energy, you stop here in the absence of ATP. You need ATP to bind, remove this, then you have this partial dissociation that primes it, so it gets it cocked and goes. So you, in rigor mortis, you stop here because once you lose this ATP, ADP, and PI, you have no more energy to get the whole system going again. So that's what rigor mortis is, and that's why it's different than relaxation. Where you have no calcium, you stop here simply because everything's ready to go but not quite ready to go. And so you're ready to contract. So cross-bridge cycling, binding, dissociation, power stroke, ATP binding, resetting of the whole system, cleavage of ATP, and going again. Calcium is what normally controls the system, and in certain pathological systems, ATP will control that system, and you will develop an excessive amount of tension or rigor mortis. So remember that at rest, the myosin is primed. So it's had a partial hydrolysis of this. So it's used some of its energy to get it all cocked, as it will, you know, like the hammer of a gun or some other thing like that. So it's used part of that energy to get it cocked, and it uses the rest of the energy when it removes it and returns to its final destination. So partial hydrolysis keeps it primed and ready for action. Think about it. That's a good thing for muscle contraction. You want it to be ready to contract when it's ready. And calcium is what initiates it by relieving suppression of it. So if it's unsuppressed, it'll just go and it'll work. But calcium has to be present because otherwise it'll be inhibited. And a change in conformation of that myosin head group is what provides that power stroke. So that's how an individual protein works and myofibrils work and how they're coordinated. So one of the other things that goes with skeletal muscle is this idea of you want to modulate force. You want to be able to recruit more myofibrils through the nervous system. You want to be able to move quickly, so you need it to relax quickly. You need to develop different levels of force. And so in skeletal muscle, you get an action potential. Now, an action potential is kind of like a 1 or a 0, a digital input. In skeletal muscle, it either fires or it doesn't fire. And so the way that muscle tension is encoded by stimulating the muscles, skeletal muscles, is by the rate at which stimuli occur. And you can see, if you get a single stimulus, you excite quickly, you relax quickly. You give multiple stimuli, and with time, these will start to sum up, but you're losing, you know, it's higher, relax, higher again, higher, relax. And so it'll start to sum up to a level, and then it relaxes very quickly as all of them relax. So this process here, you can see this summing. If you go at a very fast rate, you will develop tetanus. Not tetanus the disease, but a tetanic contraction. That means, you know, sort of a smooth, stable generation of a particular level of force. So these little bumps will average out. In fact, there are a bunch of little bumps in there, but they're, because as you see, as you get faster and faster and more, the little bumps are smaller. So this is just a process of averaging that makes it smooth. You get a nice smooth action, and it's controlled by rate of stimulation, and of course, in multifibral things, by recruitment of additional fibers. So skeletal muscle contractions are voluntary, so they're recruited by stimuli. And the way those stimuli work is that they control tension by the rate at which you stimulate them. So more stimulation, the more you have. Now this is all by itself here on the slide. The reason is that the other muscle types don't work like this. So this is one of the things that's unique about skeletal muscle that you need to know. But in cardiac muscle now is very similar in many respects to skeletal muscle, but it's different as well. It has differences in cellular organization. So cellular organization, structure, and function. So the structure is different, and therefore its functioning is different. So there are differences in the way that it's using its calcium signaling, and then it is very different in how it excites the tissue. Your heart is not the same as skeletal muscle in how it's excited. Now, we'll go over later, but your heart will beat all by itself, whereas skeletal muscle actually requires some sort of input. But the organization now at the lowest level for cardiac muscle is different. Now, you look under a microscope, you can see it right away. It's still arranged in sort of little fibrils and such, but within the myofibrils themselves, within the fibers themselves, it's not a big, single, long, multinucleated cell, all right? It actually has boundaries. Each cardiac cell is separate, and they're separate cells, and they're connected. They're connected by intercalated discs, primarily at the ends of them, so that they still line up in little trabeculae, the same way you would see in skeletal muscle, but they are separate cells. So you have a series of separate cells. Their communication with each other, both mechanical and electrical, occurs at the intercalated discs through various anchoring proteins, and electrically through connexins, which are large pores we'll talk about later. So most important difference between cardiac and skeletal muscle at one level of organization is we have individual cells in cardiac muscle. But if we look at the sarcomeres, very similar in their appearance in many respects. They have mitochondria in there, a lot of energy usage, as with all skeletal muscle. And you have that regular matrix of striations. You have Z-lines and such, and bands. But you have T-tubules, longitudinal SR, terminal cisternae. So what's different? Well, that looks pretty much the same. And sort of the physical arrangement on a gross scale is very similar. So they are comprised of individual cells. That is not the same. They're connected instead by intercalated discs. They communicate electrically through gap junction channels or connexins. And they have very similar, well-defined sarcomeric structure and an extensive SR, sarcoplasmic reticulum. Sarcomere, sarcoplasmic reticulum, sarcolemma, two terms you need to know. So that being said, then, well, before we go and talk a little bit more about it, we'll just remind you that it has its own intrinsic electrical control system and its own specialized properties. So when you beat your heart, you want it to beat in order. You have a sinoatrial node, which is your intrinsic pacemaker. When you have sick sinus syndrome, as many elderly people do, or certain other diseases, your sinoatrial node won't function properly. This is the natural pacemaker of the heart. This is what's replaced with an artificial pacemaker. So it sets the rate at which your heart beats normally. And electrical flow goes through the right atrium, left atrium, which have to contract early in order to push that last little bit of fluid into the ventricles. If they're not functioning properly, blood tends to accumulate in here. When blood stands still, it tends to clot. That's why there's a strong relationship between heart problems and stroke. So after they contract, the AV node and bundle of His are here. The AV node says, okay, wait a minute. We need to wait because we don't want the ventricles to contract because they're big muscles and they can push a lot harder than the atrium. So we want the atrium to clear that blood, delay, and then the ventricles to contract. The atrioventricular node, or AV node, is what causes the delay. Once that delay has occurred, we don't want slow conduction anymore. We want everything to excite all at once, or fairly close to all at once, so that we get a rapid propagation of that electrical impulse through the His-Purkinje system. So the bundle of His and then the bundle branches of the Purkinje system go around and they spread. And these are specialized muscle cells. They're not nervous system cells. Specialized muscle cells that spread that excitation to all regions of the heart, so you get a nice uniform contraction. As long as you don't have an abnormality like bundle branch block, in which case your one part of your ventricle contracts at a very different time than other parts and it works very inefficiently as a pump. So you need to know that there is this specialized conduction system. We'll talk even more about it later, but the heart is unique in that its control is not neuronal. It has specialized muscle cells whose function is really to originate, in the term of a sinoatrial node, originate the action potential and excitation, and to spread it in a uniform way that is optimal for the timing of the heart as a pump. And that the atrium is those chambers that are connected to the veins, and they return the heart. They beat first, and the ventricle beats second. So pump, it's the main pump. It's where the bulk of the weight of the heart is. So I said earlier that this cell organization of the sarcomeres was very, very similar between skeletal muscle and cardiac muscle on a sort of a macromolecular level. All the same sorts of things are there, but it's a little different. And you'll find that with a lot of things in the various tissues, and particularly in muscle cells. We have all the same things there. They're just organized a little differently or have different connections for their different function. So heart action potential is going to be a couple of 100 milliseconds long. It's not going to be 5 or 10 milliseconds long like a skeletal muscle action potential. It's very long. And in fact, that length, the length and shape of that helps determine the strength of contraction and its relaxation time course. So a lot of information is encoded in the shape of the cardiac action potential, as opposed to the frequency of the cardiac action potential. The frequency matters because that tells you how fast you're pumping, but it is the shape of it. And the way that shape changes things is through calcium again. Calcium is that way that we go from an electrical signal to a chemical signal. So we go from electrical to chemical through calcium. And we'll find two key differences here. First, calcium can enter through the sarcolemma. So we have sarcolemmal calcium channels that go directly into the cytosol and provide calcium for excitation and contraction directly. We still have the SR, the terminal cisternae, and the T tubules. So they're organized, as you see here, terminal cisternae, some proteins that keep them close to the T tubule. Not the same membrane, but they're in close proximity. And here you have calcium going in, and you see this arrow of calcium going this way. That is that calcium that comes in through the L-type calcium channel, or the calcium channel, contributes some of the calcium that causes excitation and also activates the sarcoplasmic reticulum ryanodine sensitive release calcium channel. So it's calcium-induced calcium release, or CICR. Calcium-induced calcium release is what causes things to go. It's a very complicated process by which the calcium comes in through the L-type channel, diffuses very rapidly across this very, very small space, binds to the ryanodine receptor, and causes it to dump calcium out of here. And then the rest of it is pretty similar to skeletal muscle. You diffuse the calcium out, goes to the troponin complex, initiates that set of contractile events, returns, is pumped back up by the sarcoplasmic reticulum, calcium ETPase, and is bound by calsequestrin here within the terminal cisternae. So very, very similar, except for this connection is very different. It's not a direct connection, it's indirect through the diffusion of calcium. And also we have calcium coming in from other sources, which of course that need also to be extruded. The calcium goes out through a sarcolemmal calcium pump and a sarcolemmal sodium calcium exchanger. And the sodium calcium exchanger pumps 1 calcium and 3 sodiums in. And so it's modulation of these processes, calcium entry and such, by this voltage in the action potential that determines the strength and of contraction and its timing of relaxation. So, important points to know. You got calcium. Calcium from the extracellular space is very important in cardiac muscle, not really so much important in skeletal muscle. And you have then a combination of pumps and exchangers in different compartments of the cell that are pumping things in, pumping things out, and those are really working together as a coordinated and very complicated system to determine the decline of that calcium transient. Remember, the cross-bridge cycling is just going to go on as long as calcium is there. These head groups won't detach and relax that muscle until such time as calcium is pumped out. So as long as you're excited, calcium is entering during that action potential, you're not going to relax that tissue. It's going to keep contracting, and so that is the direction, the reason or way that the action potential shape, that is what voltage is at, because that determines the driving force for calcium, and the duration, because that determines how long calcium can come in during that action potential, will change how hard you're contracting and how long you're contracting and how much time you have to relax and refill that heart. So your pump here is controlled through a series of nonlinear interactions of currents and subcellular stores during the action potential. And the result is something very different than you saw for the skeletal muscle in terms of behavior. We have this, instead of being over and done and a little blip here, we have a large, long duration, hundreds of milliseconds long, depending on how fast your heart rate is, action potential. During the action potential itself, calcium keeps coming in, and developing contractile force. And it's only when you repolarize in that sodium-calcium exchanger, because it's carrying current, it's got sodium, and it's got calcium. It's carrying 3 sodiums for 1 calcium. The charge on that, calcium is divalent, so it has a charge of +2. That's what that +2 at the top means. So it's carrying a charge of 2, and sodium is carrying, there are 3 of them, so they're carrying a charge of 3, so it carries a net charge of 1. That makes it a voltage-dependent process. So whether calcium is going in or going out depends on a sum between the energy of that sodium, which wants to diffuse in, and calcium, which also wants to diffuse in, but has a voltage working on it. And so that fairly complicated set of interactions between charge, voltage, because voltage is a force that operates on charge, and 2 chemical gradients that are coupled determines sort of when you can pump out that calcium from this cytosolic space And so you get this enormous delay in contraction, and you really have to return to rest before the cell starts to contract. On a larger time scale, because you have that large ranitidine sensitive release, you have an early peak and a slow decay. So that early peak and slow decay is what you see for calcium. Force, of course, is because you have to diffuse to the spot, you have to bind to it, you have to remove things. Occurs with an even greater delay, and will only start to relax when you're below a certain level of calcium for a certain amount of time anyway. So you see that the calcium transient SR release, because you get calcium in through the L-type calcium channel, you get calcium in through, it diffuses through from the T tubules into the ranitidine sensitive release, causes that release, you get a big release, and you get, then you have to pump it back up into the SR, which provides one component of this. And another component, of course, is pumping it back out through the sarcolemma. When you have neural input, you do have a neural input to the heart. That's what tells you, when you get your fight or flight reflex and your heart starts beating very fast and very pumping. That's, we call it a burst of adrenaline. So that's your beta adrenergic system going. And it's not directly stimulating contraction, but what it is doing is modifying these calcium handling properties. So the L-type calcium channel gets very, very large. The pump starts to become very active. That circa pump is regulated by isopaterenol. So it goes through, binds, it goes through a signaling cascade, activates that pump, everything else, really revs up the system. So you get a lot of calcium in, you dump a lot of calcium out, but you pump it up and you pump it out very quickly. So you get this shift both in the peak of force and also then in the development of force. So you're developing it faster, you're peaking sooner, and you're relaxing quicker because you're pumping it out of there. So these things are under neurohormonal control. It's just not direct control like tetanic contraction in skeletal muscle. So we do have inputs to it. Isopaterenol is a drug that mimics adrenaline and beta adrenergic stimulation of the heart. So the basic mechanics of heart myofilament behavior are very similar to skeletal muscle. We still have troponin, and I'm showing this again just to remind you, because if I show it several times, you pretty much know that you got to know how this works. Same sort of thing. We still have actin, we have troponin, we have tropomyosin. And these contractile elements are interacting same way. So you have head groups here, tropomyosin, buying calcium, you move it out of the way, same stoichiometry. There are differences in which proteins are in which muscle types, both within, there are different isoforms. They're basically the same proteins, but just slightly different, somewhat optimized for function in the cardiac system and in the skeletal system. So you have specific cardiac actins versus skeletal muscle actins, but basically their organization is the same. So same thing, calcium goes in, it stays up for a long time compared to skeletal muscle. So you're in this position for a very long time, and you have a long time to contract, and then you have a very long time of relaxation. And so you move up, you stay up fairly high for a very long time, and you come back down, you stay down for a very long time. And so these things are contracting and relaxing with that same cycle, cross bridge, contraction, relaxation, contraction, relaxation. So calcium comes in, binds, moves things out of the way, stays up for a very long time. So you just keep going around and around this until you develop force. And then calcium goes down, you stop here, you relax. The whole heart just, and it has to refill. That's what happens here, just as it does in skeletal muscle. So we have the exact same thing. Like I said, you really need to know those particular charts if they keep showing up, because they're a very general principle. But also then there's some other points. So cardiac action potentials don't arise from electrical stimuli, extracellular stimuli, normally. There are exceptions to this. When you see somebody go into a heart attack and they shock them, that's an extracellular stimuli that can cause it. Sometimes if people get a huge discharge of their neurons, they can cause an ectopic beat in the heart. So you can elicit you can use an extracellular stimuli to elicit a cardiac contraction. That's why defibrillators work, but they do not normally arise from extracellular stimuli, and they don't sum and produce tetanus. Tetanic contraction in the heart would be kind of a bad thing. You either want it to be pumping or not pumping. Pumping or filling, that's all it wants to do. If it goes into tetanus, it's not pumping. And so it's very, very different. You don't sum the action potentials and they don't go into tetanus. If you get multiple action potentials at too high a rate, some very bad things happen to you. And we will talk about that probably in lecture 4 or 5. Cardiac contraction is dependent on not only release from the circumplasmic reticulum, but on external calcium entry through L-type calcium channels. And it has the important thing of calcium-induced calcium release. So cardiac action potentials have that. So they're controlled much more by the shape, size of the surface action potential, and particularly through calcium entry and exit during the various phases of the cycle. Smooth muscle. Now, I can't really do justice to smooth muscle in the time allotted. It is really not one thing. There are a lot of different types of smooth muscle all over in your body. They're in your viscera, so your digestive system, your intestines, your stomach. They're, for half of the class anyway, they're in your uterus, uterine muscle. They're line many of the ducts and veins in your heart. And because they need to contract for different lengths of time and or in different ways, their contractile behavior and their control of contractile behavior is really variable. You could spend easily a whole course on smooth muscle types, variability, and how they function. But it is important to know about it and sort of know at least some of the generalities of it. Why is it called smooth muscles? Well, if you look at these pictures of smooth muscle, and we have these smooth muscle cells organized in different things from your textbook here, what's the thing you notice about it? It doesn't have striations. So it doesn't have these ordered bundles, and it doesn't have these ordered arrays of contractile elements. Hence, it was named a long time ago smooth muscle, because it looks smooth. It doesn't look rough or corrugated. So smooth muscle, pretty much what you would think. It looks smooth. Well, that's not really terribly informative, is it? So when you have smooth muscle, it does have organization. There's this picture from your textbook again showing this organization, and this organization looks a lot more regular than it actually is. It's fairly irregular in these respects, but it does make some points, important points about commonalities. We have gap junctions. Now, gap junction proteins here, there are a large number of different gap junction proteins, and they may be different than the ones in heart, but basically they're the same thing. They provide electrical coupling between the cells. So, like cardiac muscle, your smooth muscle cells are organized in cellular format. So they're individual cells, and they work as a syncytium, same as the heart. A syncytium meaning same cell-like. They are separate cells, but they're electrically connected in order to act in a coordinated fashion and mechanically connected. And so we have these dense membrane areas with different sorts of connective proteins, which actually will span this gap here, providing mechanical connections. And you can see that the contractile filaments are actually anchored to them. And what do we see there? Thin and thick filaments. So thin and thick filaments are still active. And it's still the sliding of thin and thick filaments in the smooth muscle, but it's different. So it's the same but different from that point of view. Smooth muscle still works using smooth muscle sliding filaments, but these aren't optimized in terms of being really big ordered arrays. They tend to be diffuse through the whole cell. They may be specialized in their organization depending on the tissue that they have. So we'll start now at the level of extracellular to intracellular communication and what happens there. Well, calcium is still at the center of it. Calcium is a pretty important signaling pathway. It's really the change by which we go from electrical activity to chemical activity. But we got a lot more arrows in this one. We still have a sarcoplasmic reticulum, so this intracellular store here that is now not so well organized. It's just there. And it still uptakes and releases calcium. It has a circuit pump, so that's that ATPase-dependent pump takes up calcium into the SR and prepares it for release. Now, it can have ryanodine-sensitive release channels, just like skeletal muscle, but notice that they are not in these organized T-tubule type of things. You have to diffuse there, and then you have to bind calcium and have calcium-induced calcium release. So very different in its organization. It's not spatially organized. Ryanodine receptor release, not all cells may have it of the different types, so keep that in mind. It's not every smooth muscle cell has everything, but in general, they can have these things. Also equally important is the IP3, or inositol trisphosphate gated channel, and that goes from a chemical input, from a hormone or a neurotransmitter, and activates IP3, or inositol trisphosphate, and that goes in, it binds to a receptor, and this opens up a different type of calcium release channel from the SR. So the calcium can come in from the calcium release channel. Calcium can still come in through the cell surface calcium channels, and there are a lot of different types of these, varied for all these. And of course, when that happens, you still need a pump and or a sodium calcium exchanger to get that out. And it's still involved heavily in contraction. But it's a little different. So let's go over the things that are different at the cellular organization level before we get to that difference. So SR calcium release, still very important in smooth muscle. Calcium from the extracellular medium, also important. IP3 calcium, also very important. So direct hormonal regulation of contraction, it can occur. And many of the same uptake and release mechanisms in a general sense are held in common with the other types of muscle. We still have our sarcoplasmic reticulum ATPase pump. We still have a sodium calcium exchanger. We still have calcium channels. We still have ryanodine sensitive release channels. So it's just a more complicated, more general system. And one of the things that's also more distinct about this is that we no longer have calcium binding directly to troponin and activating the troponin-myosin complex and troponin C. Instead, we have a much different mechanism. So what calcium does, we still have 4 calciums go in. They have to bind to a calmodulin myosin complex, or myosin kinase complex, excuse me. So they bind to an enzyme. Calcium goes in, binds to calmodulin, binds to an enzyme. That enzyme is a kinase. So it phosphorylates the channel. So it puts a phosphate group on. And phosphate goes on at a regulatory site. And what does that do? Well, that allows the whole system to proceed. Now, here, if that phosphate comes off, you have phosphatase. You have these constitutively activated phosphatases. If that phosphate comes off, the whole system won't work. It's just relaxed and it's going to stay there. It's ready to work, but it needs that phosphorylation there. And so when you phosphorylate it, it all of a sudden becomes active. So instead of moving troponin-myosin complex out of the way, tropomyosin complex out of the way, you activate this, and it just starts cycling around, same as before. You end up in Riger down here, if you don't have any ATP, to unbind the head group, reset the whole process. But you don't need any calcium here. Calcium goes in here, and instead regulates an enzyme. And that has some of advantages. But the important thing to remember, in the smooth muscle, it's myosin kinase, not a tropomyosin troponin interaction. Okay, so completely different mode of regulation, and that gives you some advantages when you're a smooth muscle. Smooth muscle cells, in general, for a lot of purposes, don't have to respond very quickly. Sometimes they set up, sphincter muscles want to remain contracted all the time. You don't particularly want to spend a lot of energy on them. So this provides you with a lot of ways to regulate it, especially because this phosphate can be removed at any point in the cycle. And if it does, you can be stuck in a contracted state, a relaxed state, or some intermediate state. And so there are 2 types of contraction generally, although really I think it's a continuum of them. You have phasic contraction and tonic contraction, where you get a stimulus, you phosphorylate things, it cross cycles, you get some force, and it's transient. Transient means it's over and done with. Phasic basically means the same thing. It's in phase with repetitive stimulation. But similar to skeletal muscle, you can develop tetanus. Now, rather than stimulate it all the time, one of the things you can do is phosphorylate them and then stop it in a certain level. And that gives you tonic contraction or steady state contraction, and it just stays contracted. So just to summarize, because I know you got to go to class now, skeletal muscle, SR calcium release, directive repetitive stimulation. Those are important things. Cardiac, extracellular calcium, action potential, shape, and duration. Smooth muscle, kinase and IP3. Those are the key and important differences. Thank you very much. Hi.
I have a couple questions about smooth muscle.
Certainly.
You were saying something about the syncytium?
Syncytium, yes.
What is that?
Syncytium, same cell.
Okay.
So
But that's not
we call things a functional syncytium, meaning that even though there are cellular boundaries in them, they're so well connected that they functionally are the same.
because of the gap junctions.
Right.
Okay. And then you said the SR of the smooth muscle is less organized. I mean, does it not have cisternae, or is it still has that?
No.
It doesn't.
Well, I don't know in all cases because it's really varied, but in general, no, it doesn't have.
Okay.
It doesn't have that longitudinal stuff. It doesn't have the terminal cisternae. It may have some levels of organization, but nothing that generalizes the way that.
It's not as complex.
Right.
Okay. Great. That's all. Thank you.
Alright, so I was
Hi.
hoping you could clear this up for me here.
Certainly.
For this slide, I thought you said that a lot of cells have the same things, but they're organized differently.
Right.
But here I wrote down that there'll be different isoforms of the protein, but the organization is similar. Is either of those right or incorrect?
They're both correct.
Okay.
Okay. An isoform, there are a lot of different troponins. You got troponin, smooth muscle troponin. You got smooth muscle actin. You have atrial specific beta myosin heavy chain. So each one of these proteins is the same from this general categorization. But when you get right down to it, if you were going to look at it on a gel, they would have slightly different molecular weights from different tissues, and certain differences in sequence. But generally they have the same structure, function, and organization.
Okay. I mean, at this level,
at this level,
as opposed to that structure, rather?
Well, so this has certain structural organization here, but it's different from skeletal muscle in that the structure differs here. There is no structure spanning that across that transduces the calcium.
Okay.
The structure is different. These dihydropyridine receptors are functional channels. The structure is different because these channels are out here.
Okay.
So those would be several of the main differences. Differences in isoform, it's pretty much true of all organs, so that you have to check which isoform is in what organ. People need to know that those things occur, because if they don't, if you run a gel and you're looking for the wrong molecular weight, because you're getting the tissue from the wrong place, you just need to know that.
Right.
But functionally, in terms of general principles, they aren't really different.
Okay.
The myosin is obviously different than the smooth muscle because it has that phosphorylation regulation site, which skeletal muscle does not.
All right. Cool. Thank you.
You're welcome.
I saw your assigned readings online. Do they correspond with your lectures, or they jump around? Just one.
They're more or less in the same general order in which I'm going to be doing things.
Right.
I didn't specify them per lecture.
Right. Yeah.
So, but they're in the same general order.
Okay. Thank you.
Do you have assignments online?
No, I posted the lectures, the notes online,
Oh, okay, so what was
and assigned readings.
Oh, assigned readings.
Yes, there are assigned readings posted online.
Okay. I wanted to ask a general question about this. Are you going to go into detail in it further?
Yeah.
Okay, so you were just setting the foundation.
Yeah, you see these things and you go look at the molecular mechanisms of what's going on, and then you go back and so we sort of start out, with things people are familiar with, pumping, and talk about how we use these in grafts, and go down to a molecular level, and then we come back up to macromolecular organization as we go back up, and then so we cover a lot of the same turf, but from a different, from an organization, reductionist approach versus an integrative approach, sort of over the course of the lecture.
Okay. And then in the beginning, you had the distinction. This is the skeletal muscle, right?
That's what?
Skeletal muscle.
Yes.
And then you have the cardiac muscle back here. Do you want us to know the distinctions of like the cycle?
Well, yeah.
Okay.
Yeah, I don't know what's different.
What's the main distinction here when you said that it's released from the T-type?
Calcium-induced calcium release. I'll hit this again several times. Calcium-induced calcium release, not direct release. Entry of calcium from the extracellular space and extrusion of that extracellular calcium.
Is that from the L-type channel?
Right. In the cardiac muscle it is.
Okay, and then it goes to the ryanodine receptor and it